Hello, my name is Kevin Fuller, and I'm an assistant professor in the Department of Ophthalmology, as well as in the Department of Microbiology and Immunology at the University of Oklahoma Health Sciences Center, located in Oklahoma City. I'm specifically located at the Dean McGee Eye Institute, which is both a comprehensive eye clinic, as well as home to a variety of vision research programs that study diseases ranging from macular degeneration to glaucoma, to the diseases that my lab is interested in, which are fungal infections of the eye, specifically the cornea. So unfortunately, fungal corneal infections, also called fungal keratitis, are about as bad as they sound. Conventional therapy in the form of antifungal drops fails about 50% of these patients, which means they require at least one and sometimes repeated corneal transplants to save vision in that eye. And if the infection can't be cleared either medically or surgically, then the entire eye sadly has to be removed. And there are an estimated 1 million cases of fungal keratitis worldwide and about 30,000 cases in the United States. And there are really two major risk groups associated with these infections. The first is contact lens wearers, which of course is a very large demographic in the United States, and one that encompasses effectively all socioeconomic groups. And indeed, there was an outbreak of fungal keratitis in about 2006 that was associated with a contact lens cleaning solution. And despite the quality of care you would expect in this country, about a third of those patients in the outbreak required a corneal transplant. But perhaps the largest group at risk for fungal keratitis are agricultural laborers or farmers that routinely experience corneal abrasions in the field due to vegetative debris. So vegetation, as you would imagine, is highly associated with fungal organisms. And so that means fungal keratitis disproportionately affects low-income individuals that might have limited access to quality health care, let alone quality vision care. And so my research program is focused on better understanding the pathology of fungal keratitis in order to develop better treatments, preferably non-surgical treatments, that will mitigate the quality of life and economic burden that these infections impose. So this image shows a healthy cornea, which is normally clear to allow light through to facilitate vision. The bottom image is a fungal keratitis cornea, and we understand that the damage that occurs to the corneal tissue during infection is a result of two things. The first is the fungus growing there, which of course can directly cause damage to the tissue. And the other is the massive inflammation that occurs, so immune cells being recruited to the cornea from the bloodstream that occur in response to the fungus. This leads to corneal opacity and the formation of ulcers that are painful and can result in scarring and long-term vision loss. Accordingly, an important aspect of managing fungal keratitis is managing that inflammation. So one of our projects seeks to better understand how this inflammation is initiated. More specifically, we're interested in cells that are already there in the cornea that can both recognize the fungus and initiate this inflammatory cascade. One of the major cell types present in the cornea that have not been explored in this regard are corneal fibroblasts, whose job is normally to secrete and build the structure of the cornea itself. So to study these cells, we have developed a model whereby fibroblasts are isolated from human donor corneas and then plated onto nylon membranes in a petri dish. And after about a month or so of incubation, the cells will have done what they normally do, which is secrete collagen and generate a 3D matrix. In other words, what we'll have is a human corneal stroma in a dish. So the bottom left image shows one of these constructs where the fibroblasts, labeled HCFs for human corneal fibroblasts, are present within a dense extracellular matrix, labeled ECM, which is primarily comprised of collagen, all resting on top of that nylon membrane that I mentioned, labeled M. We then infect these constructs with our fungus, and we can see what we would expect to see, which is damage to the collagen matrix itself associated with fungus growing throughout. And the fungus can be seen in that lower panel stained black. So shown here are electron microscope images that clearly and remarkably, we think, demonstrate fungal cells or spores being internalized within the fibroblast, again labeled as HCFs. And in some panels, it actually looks like the HCFs are shooting out these projections, kind of like a lasso on a rope, in order to take in the fungus. And the ability of corneal fibroblasts to do this, that is to internalize microbes, has not previously been described, so we're very excited about these observations. So we have also shown, although not illustrated here, that after the fibroblasts interact with the fungus, they secrete these pro-inflammatory molecules called cytokines, which would then play a key role in recruiting white blood cells or immune cells into the cornea and drive that inflammatory response. So we're following up with this study to identify molecules on the fibroblasts that drive this activity and could be potential targets for new drugs. So for example, if a patient came into the clinic with a corneal abrasion and was deemed to be at high risk for developing fungal keratitis, then they could be given this drug to block the development of that damaging inflammation right from the beginning. So moreover, we think that the model that we've described here has a lot of utility. So we have several projects in the lab that utilize this model, and we're very excited about them. So I really appreciate your time and interest in our research. Being part of the EVS advocacy program is a real privilege and a real pleasure. So I thank you.